In this fourth and final screencast for Chapter 2, we'll look at three families of organic molecules: lipids, proteins, and nucleic acids. Like carbohydrates, most lipids are made only from the elements carbon, hydrogen, and oxygen. They are different from carbohydrates in that the ratio of these three elements is far from 1 to 2 to 1. Lipids have much more carbon and hydrogen and very little oxygen. Unlike carbohydrates, lipids have little or no hydroxyl groups. They have very few to no polar covalent bonds. Therefore, they're hydrophobic. We'll look at four different kinds of lipids. The simplest are the fatty acids. Glycerides and phospholipids are made from fatty acids. And then the final group, the steroids, are sometimes called the non-fatty acid lipids because they're not synthesized simply from fatty acids. So let's start with fatty acids. They're called acids because they have an acidic carboxyl group, the COOH, on one end of the molecule. The rest of the molecule is composed of only carbons and hydrogens. This part of the molecule is called a hydrocarbon chain. The carbon to carbon and carbon to hydrogen bonds are all nonpolar covalent bonds. More specifically, the fatty acid pictured here is a saturated fatty acid. By saturated, chemists mean that every carbon atom in the hydrocarbon chain has two or more hydrogen atoms bonded to it. Saturated fatty acids have a hydrocarbon chain that is nice and straight. These straight molecules stack tightly together. This facilitates them being a solid at room temperature. Saturated fatty acids are common in animal fats. The second fatty acid is unsaturated. What this means is that two or more of the carbon atoms have only one hydrogen atom bonded to them. They have only one hydrogen atom because the carbons are double covalently bonded to each other. In addition to reducing the number of hydrogens present, the carbon-to-carbon double bond puts a bend in the molecule, changing its shape. Because of the bent hydrocarbon chain, unsaturated fatty acids don't stack easily together. As a result, they tend to be liquid at room temperature. They're commonly found in vegetable oils. Trans fats are partially hydrogenated vegetable oils. Processed foods often contain partially hydrogenated vegetable oils because they're more stable. Glycerides are lipids that are made from glycerol and 1, 2, or 3 fatty acids. Monoglycerides contain 1 fatty acid, diglycerides have 2 fatty acids, and triglycerides have 3. Let's look at how they're synthesized and how they're broken down during digestion. In the green box is glycerol. Using condensation synthesis, the same kind of reaction used to make polysaccharides, 1, 2, or 3 fatty acids can be bonded to the glycerol. A molecule of water is released when each fatty acid is attached. Hydrolysis of glycerides is also possible. This occurs during the digestion of these molecules. The making of lye soap is another example of hydrolysis of glycerides. Your great-great-grandmother may have taken animal lard, which is rich in triglycerides, heated it in a large kettle with water and lye to make soap. She was hydrolyzing fatty acids from the lard. Earlier, I emphasized that the hydrocarbon chain forming the bulk of a fatty acid molecule is strongly hydrophobic, but there is a polar covalent bond between the oxygen and hydrogen atoms in the carboxyl group. This part of the molecule will form a hydrogen bond with water and therefore is hydrophilic. Emulsifiers, like soap, allow water and lipids to mix because the fatty acid mixes well with both substances. The hydrocarbon chain mixes well with other lipids, and the OH of the carboxyl group attracts water molecules. A third type of lipid is the phospholipids. Earlier, I said most lipids contain only carbon, hydrogen, and oxygen. I couldn't say all because of the phospholipids. They contain carbon, hydrogen, oxygen, phosphorus, and some other elements. Much of the phospholipid molecule is a diglyceride. It does contain only carbon, hydrogen, and oxygen, and is strongly hydrophobic. However, the other end of the molecule has a phosphate group with something else attached to it. When chemists want to indicate that something else is present, but they don't want to specify what that component is, they'll often use the abbreviation R. The phosphate containing end of the phospholipid is strongly hydrophilic. Phospholipids are important because they form the basic structure of all cell membranes. Here's an illustration showing a phospholipid. The hydrophobic part is shown in orange, that's our diglyceride, and then much of what you see in red is the hydrophilic part, which contains the phosphate group. The last type of lipid we'll discuss are the steroids. They're sometimes referred to as non-fatty acid lipids because they're not made by simply attaching fatty acids to something. Like other lipids, they contain lots of carbon and hydrogen and very little oxygen. They're hydrophobic, like other lipids. Cholesterol is a steroid which you probably have heard the most about. While too much can cause health problems, cholesterol is absolutely necessary as a component of animal cell membranes, and it's also a precursor to the sex hormones estrogen and testosterone. In this diagram, you can see the similarity between cholesterol above and testosterone below. The third family of organic molecules I'd like to discuss are the proteins. Unlike carbohydrates and lipids, proteins contain a lot of the element nitrogen. The nitrogen is present in the form of amino groups. Proteins are polymers made from long chains of amino acids. There are more different kinds of proteins than any other type of molecule in your body. As you might guess from the diversity of kinds of proteins, they play a wide variety of different roles in your body. Among proteins' functions are structural, movement, and functioning as enzymes. As we saw with starch and cellulose, shape is very important in determining the physical and chemical properties of organic molecules. This is certainly true for proteins. Proteins are polymers of amino acids. So, what is an amino acid? As the name indicates, at one end of the molecule is a nitrogen containing amino group. Bonded to the same carbon atom, but opposite the amino group, is the acidic carboxyl group. All amino acids share the same basic structure: a central carbon atom, which has an amino group attached to one side, and the acidic carboxyl group at the other. How amino acids differ is what else is attached to the central carbon atom. It can be as simple as a hydrogen atom or much more complex. It's been estimated that there are well over 1 million different proteins in the human body. All of these proteins, and all that we know about in other living things, are made from the same 20 amino acids. Of these 20 amino acids, 9 are identified as essential amino acids for humans. By essential, what we mean is that they're required, but cannot be synthesized by your body, and therefore they must be present in your diet. These are a few examples of amino acids. Notice the amino and the acidic carboxyl group attached to the carbon atom at the top of each. They differ in what is present below in that pinkish or red box. Small sequences of amino acids are called polypeptides. Poly means many. Peptides refer to the chemical bond that links each amino acid to its neighbor. More specifically, peptide bonds are the covalent bond joining the carboxyl group of the first amino acid to the amino group of the following amino acid. Guess what? You're already familiar with the chemical reactions used to synthesize and also to digest polypeptides in proteins. Our old friend condensation synthesis is used to create peptide bonds between two amino acids. An OH from the carboxyl group of one amino acid and a hydrogen from the amino group of the second amino acid are split off and form a molecule of water. A new covalent bond then forms between the carbon and the nitrogen atoms. It's this bond that we call the peptide bond. Plant and animal proteins in your diet are broken down using hydrolysis. A molecule of water is inserted, breaking the peptide bond. Denaturation is a change in the shape of a molecule. Changing the shape of a molecule can have profound effects on its chemical and physical properties. Let's look at two examples. Protein is abundant in egg white. When raw, egg white is a liquid, nearly colorless, and transparent. When cooked, the proteins denature, changing shape. The change in shape causes the egg white to become a solid. It changes to a white color and becomes opaque. In this example, the denaturation is permanent. In sickle cell anemia, the protein hemoglobin, which is found in red blood cells, changes shape. In the presence of oxygen gas, the protein is donut-shaped. When oxygen is removed, the protein becomes more sickle-shaped. This change in shape of the protein also changes the shape of the red blood cells. They go from looking like jelly donuts to jagged, somewhat sickle-shaped cells. In this case, the denaturation is easily reversed by adding oxygen. Our fourth and final family of organic molecules are the nucleic acids. They're polymers made from nucleotides. DNA and RNA are examples of nucleic acids. Like amino acids, nucleotides contain the element nitrogen. Unlike amino acids, the nitrogen is present in the form of a 5 or 6-sided ring called a nitrogenous base. These nitrogen-containing bases are attached to a 5-carbon sugar. Attached to the other end of the sugar is one or more phosphate groups. This diagram represents a simplified picture of a nucleotide. In blue is the nitrogenous base. The green pentagon represents the 5-carbon sugar, and the yellow circle with a P in it is the phosphate group. Only 4 different nucleotides are used to make all DNA. These nucleotides differ in which nitrogenous base is present. This represents a close look at a short segment of DNA. In the box is one nucleotide with thymine as its nitrogenous base. The other bases are adenine, guanine, and cytosine. Notice that A and T, adenine and thymine, will form hydrogen bonds with each other, and so will cytosine and guanine, C and G. A nucleotide which you may get tired of hearing about is ATP, adenosine triphosphate. Unlike the nucleotides that make up DNA, ATP has 3 phosphate groups. ATP is an important molecule in the bodies of living organisms because it supplies energy. Here's a simplified picture of ATP. In the blue is the nitrogenous base, then the 5-carbon sugar, and 3 phosphate groups. It's adding or releasing that 3rd phosphate group that allows the molecule to store a lot of energy. Don't worry about the fact that the nitrogenous base is on the left-hand side here. They just simply rotated the molecule for this picture. That completes Chapter 2.